So we'll move on to our next speaker. Dr. RNA Arias is going to be our next presenter. She is a member of the graduate faculty of the Plant Pathology Department at UAV and has been since UGA and has been since 2012. She is also a research pathologist for the USDA and has been there since 2011. She received her BS and MS degree from the Universidad Nacional del Santiago del Estero in Argentina, and also received an MS and a PhD in plant pathology from the University of Hawaii. She is a recipient of the Presidential Early Career Scientist Award in 2014, as well as the Early Career Scientist of the Year in 2013. She's going to talk to us today about advances in RNA interference technology for the control of aflatoxin in peanut. So, Dr. Arias, if you would share your screen.
Well, first of all, thank you very much, John, for the introduction. And I want to thank the amazing team that put this together and that has been so patient with us to make it happen. Today's talk is going to be about work we are doing at the National Peanut Research Lab in Dawson, Georgia, regarding RNA interference to control aflatoxins in peanut. Now, first, I want to do a review of the general idea of how RNA interference works. Let's imagine that this is a plant cell. Here is the nucleus. And if we apply double-strand RNA to that plant cell, that will send a warning signal that will activate the RNA interference machinery, starting with dicer, which is a molecule that will cut that double-strand RNA into small interfering RNAs. And that will recruit additional molecules to form the risk factor. And that complex, if it finds a messenger RNA that matches that particular sequence, will destroy it. So any messenger RNA that happens to be in that cell and matches these fragments will be destroyed. Now, in addition, in 2008 and 2011 were the first papers that showed that if a fungus becomes in contact with the plant cell carrying signals for a messenger RNA that is in that fungus, that signal can destroy the messenger RNA in that fungus. So the idea is that if we put one double-strand RNA in a plant cell, we use the machinery of the plant cell to create these silencing signals. When the fungus becomes in contact with that plant cell, that particular gene will not be expressed. I divided this talk in 3 different sections. The first one was the proof of concept. It was a long stretch from papers that had been just published in 2011 to jump to control aflatoxins using this system. There was no evidence for it. So we had to do it using genetic transformation of peanut. The second part is finding the proper targets for RNAi. That means knowing what aspergillus are colonizing the peanut seeds, particularly in Georgia, and find those consensus sequences in their DNA. And the third part is the exogenous application of RNAi, as we call it, non-transgenic delivery to prevent aflatoxins. So we go on the first one, which was that proof of concept. Let's go back to the same diagram that we had. So now, if we have a transgenic plant, most of the cells in that plant, all or most of them, they will be expressing a double strand that we applied, but that will come from the nucleus. So we have to understand that in this case, most of the cells in that plant will be, let's say, protected against that particular gene that we want to silence in the fungus. The first step was to decide what to put as a double-strand RNA. So we knew what the genes that are being used by Aspergillus to make aflatoxins is a set of about 25 genes. So we chose 3 of them, plus another couple that help in the mycotoxin processing. We put fragments of those genes as inverted repeats in a plasmid DNA that we call P5X CAPD, and then was in the hands of Dr. Dang to do genetic transformation of peanuts. Once we obtained plants that consistently were positive as PCR for our insert, the intended insert that we wanted to put as double-strand RNA, we had to test those plants. Now, at the time, we didn't have a way to test, and besides, these plants were generating probably 2 or 3 seeds each plant. So we came up with this idea. We surface disinfect the seeds, then we remove the testa because the testa has antimicrobial compounds and we didn't want that to mask what we were after. We also remove the embryonal axis because that one produces a lot of phytoalexins that are also antimicrobial compounds. So in what was left, the 2 cotyledon pieces in the seed, we cut them in half, blotted the excess of water, and placed them in water agar with the cut part to the top. There we applied a known amount of spores of Aspergillus and incubate them to the perfect conditions to accumulate aflatoxin at 30 degrees and in these moist chambers. At 24 and 48 hours, we extracted RNA and sequenced the small RNAs. At 72 hours, and at the beginning we were also testing at 96 hours, but the seeds were disintegrated by 96 hours with this load of spores. And Dr. Sobolev did test each of these quarter seeds using UPLC and LCMS for aflatoxin. The first 3 transformation events, when we tested them this way, so we are giving them a large load of spores in the perfect conditions for aflatoxin accumulation. 3 transformation events had between 70 and 100% less aflatoxins. So between 6,000 in the control and 0 to 2 ppb in those lines. In additional 2 lines, 2 transformation events, this is work that was continued by Dr. Power. Many of you know her, and this was with the support of USAID and the Peanut Mycotoxin Innovation Lab. She also completed the analysis of a small RNA sequencing and discovered 10 novel microRNAs that were being produced in these plants. We have to pay attention to these microRNAs because we know that small RNAs can be produced for non-target, so it's unintended targets. But the results that we have up to here, we consider they were sufficient to believe that this was doable, that the use of RNAi to control aflatoxins in peanut was doable. Before, and I had a couple more comments here, is that we did test these seeds for different maturities, and the response was different. In all of them, we observed reduction in aflatoxin, but we had better control of aflatoxins in immature seeds than in mature seeds. Before we move to the next topic, I wanted to briefly mention that we have used this method of screening of individual seeds to test different peanut germplasm. In this particular case was wild peanuts, to identify those that do not accumulate aflatoxin. And the method has been very consistent in the results. So the second topic is the survey of Aspergillus. We had to find what are the most common Aspergillus colonizing peanut seeds in Georgia. and to find what sequences should we target in those particular aspergillus. So we isolated a large number of aspergillus, developed indel markers, insertion deletion markers within the aflatoxin synthesis cluster, use capillary electrophoresis to genotype each of those isolates, and perform cluster analysis with the indels. There we separated very clearly with these indels the aspergillus that were species flavus, the parasiticus, and the aspergillus flavus that were non-producers of aflatoxin. Here took over Dr. Faustinelli, working as a postdoc in my lab, also with the help of USAID and PMIL, and did the whole genome sequencing of 10 selected isolates that were representative from each of the main clades. The interesting thing was that after doing cluster analysis, both topologies match, the ones done with indels and the one with whole genome sequencing. Why is this important? Is because we can use those indels to screen large number of aspergillus, find which ones are the predominant genotypes, and only sequence those to develop targets for RNAi. The aspergillus that we identify as the most abundant colonizing peanut seeds in Georgia, we sequence them, and those sequences are available in the gene bank. I will mention other work that we have been doing with African countries before we move to the next topic. We work with isolates from Ethiopia. This is work that was done by Abdi Mohammed while he was a PhD student. Now I can proudly say that he finished his PhD and he was hired as a professor in Haramaya University in Ethiopia. He published part of the work, and we recently published 16 genomes of those isolates. The work in Uganda was led by Amos Akur, supported by NARO, National Agriculture Research Organization from Uganda, while he was a master's student, and he graduated also last year. His paper, I would say, is almost accepted because he submitted the corrections last week. Now, moving to the third topic, the exogenous application of RNAi to the peanut plant, or non-transgenic delivery. We go back again to the same diagram of the beginning. If we apply double-strand RNA to a plant cell, the only cells that will activate the machinery of RNA interference are the ones that receive that double-strand RNA. So it could be few cells in that entire plant. So now we are not only asking the plant to use the machinery to generate these silencing signals, but we expect it to move it to the neighbor cells, and from there to the phloem, and from the phloem all the way to the seeds, and accumulate in the seeds to protect them from aspergillus when they become infected. So as you see, it's a little bit orders of magnitude more complicated than working with transformation. So what were our questions at the very beginning? There was no information on peanuts. Pretty much all the work was generated from our lab in this subject. So if we apply double-strand RNA to the peanut plant exogenously, will it reach the flowers? Will it reach the pegs? Will it move at all? Or if we apply it to a particular location, will it multiply in that tissue? And if it reaches the pegs and then the seeds, will it accumulate in enough quantities to prevent aflatoxin accumulation when aspergillus infects? And what is that level? So to answer some of the questions, Dr. Faustinelli started the work in my lab working as a postdoc, also with the help of USAID and PMIL. And working in tissue culture plants of peanut, apply double-strand RNA and monitor the movement of that double-strand RNA. To have an idea what this type of experiments imply is at each sampling time, you have to cut the plant in sections of 1 centimeter, and from each piece extract RNA and run real-time PCR for detecting the fragments that were inserted. It's a lot of work. So what she found was that when applying low concentrations of these double-strand RNAs at 30 days and at 60 days, she detected them in the flower buds of peanut. With a bit higher concentration, she found them within 30 days in the pegs that were being formed. And with even a higher concentration, at 15 and at 30 days, she found both sense and antisense of these fragments in the forming pegs. Now, I will move to another question that we had. If we apply double-strand RNA to the plant, will it multiply in that location? So using the same construct that we had made at the beginning, we prepared double-strand RNA with fragments of 5 genes and plasmid DNA carrying inverted repeats of those same fragments. We used the biolistic method or GenGAN to apply it to tissue culture peanut plants and put them back in culture for 24 and 48 hours. At those times, we extracted RNA and had it sequenced. So what we see here is on the left is the application of double-strand RNA, on the right is the plasmid DNA carrying the same construct. And in colors, these lines in colors are the reads or small RNAs that were sequenced, and they were found to match the genes that we had applied. We have several observations here. First is that not all the genes, gene fragments will generate small RNAs. That also that when we apply plasmid DNA, we obtain larger amounts and also both in sense and antisense sequences. At 48 hours, we had actually for both, for double-strand RNA and for plasmid DNA, these signals, these small RNAs had double in all the treatments, but still it was more abundance in the plasmid with inverted repeats than in the double-strand RNA. In addition, because we use small RNA sequencing, we were able to analyze microRNAs that might not be our targets, but could be affected. And here we found 2 particular microRNAs that are related to plant disease resistance that were significantly affected. So in all these works, we have to make sure that we are not unbalancing other part of the plant. In the last set of experiments that I will talk about is the application of double-strand RNA in our environmental plots. Here, what we did was to apply double-strand RNA of a variety of mixtures and constructs, but I will show only one of those experiments in which we observed after we collected the seeds from those plants that were treated with double-strand RNA, and 5 weeks after harvest, we did the seed challenging of those seeds using the same method that I had mentioned before. The seeds of plants that were treated with double-strand RNA for PKSA had a 40% less aflatoxin B1, and that was statistically significant. We are still analyzing a large number of libraries that we sequence for the small RNAs, This is only one example. I don't know if it's visible in the slide, but the small RNA sequencing from seeds coming from plants that were treated with PKSA had a larger abundance of small RNAs matching the gene in Aspergillus that was our target than in the controls. We have a large number of questions that we need to answer with this analysis, and it takes a lot of effort and time. So in summary, what do we know now? We know that if we apply small RNAs, double-strand RNA to the aerial part of the peanut plant, it will reach the flowers, it will reach the elongating pegs, and we know that they will multiply in place even to reach a double within 24 hours. So after 24 hours of inoculation, in another 24 hours will double the number of these RNAs. That means they are multiplying inside the plant cells. What questions remain? Well, these are some of them. What is an effective method to deliver this double-strand RNA? Actually, tomorrow we are starting applications using a new method in our environmental plots. What is the number of applications? What concentration? What tissue to apply it? Other important question is, will our treatment reach all the seeds in that plant? And what is the minimum amount of small interfering RNAs that we need to accumulate in a seed to protect it against the invasion of Aspergillus and against the accumulation of aflatoxin, let's say. And other important one is if we can achieve that, for how long will that effect last after harvest? As I mentioned before, we tested seeds 5 weeks after harvest. We need to determine for how long this protection will stay in the seed after that seed goes to storage and in what conditions. So with that, thank you everybody. And again, to the organizers that were amazing. And I will answer any question if there is time for it.
Okay, I think we have time for one question. Do you expect that there is a difference, different toxigenic isolates that infect and contaminate peanuts grown in Georgia from those that infect and contaminate peanuts grown in other areas?
Yes, because we have seen, in particular, we didn't finish the work on Alabama. We had a large number of isolates from Alabama too. But the work that we have seen between here and Ethiopia and Uganda, the fungi are totally different.
Okay, well, great. Well, thank you, Renee. That's very, very intriguing work and sounds very promising. Thank you for that presentation. Next question is for Renee. Renee, how did you confirm the RNAi technique you targeted? Your target genes completely, partially silenced, question mark.
Well, at the beginning, we only use theoretical design by using the Argonaut software, try to find in dicer substrate software in our designs. We tried to do a genetic transformation of Aspergillus in our lab. A postdoc was working here, and he was not successful in doing transformations, so we didn't have transgenic Aspergillus to demonstrate that. We did try, we have a lot of tests done with different media where Paola working in our lab, she showed in one medium there was a lower expression of the genes when applying the various types of double-strand RNA. But it was not done in transgenics. If if Emran has a system to genetically transform Aspergillus, maybe we could collaborate and have a better design of these double-strand RNAs.